My name is Paul Hallenbeck. I'm the founder, chief scientific officer, and president of Seneca Therapeutics. This company was founded on earlier promising data generated on a novel oncolytic virus called Seneca Valley virus that was discovered serendipitously, I would add, by my team while at Novartis Pharmaceuticals in the year 2000. Great story, but for another day. SVV-001 is the specific Seneca Valley virus we discovered and is in clade 1 of the Seneca virus genus of the picornavirus family. It was initially developed through several clinical trials in the 2000s. These trials were all initiated and mostly completed before the most recent immuno-oncology revolution began with approval of checkpoint inhibitors and the immune RECIST criteria. SVV has been in over 75 patients utilizing a single systemic dose as a monotherapy. Only one DLT was observed, which was tumor pain that was resolved by analgesics. We saw encouraging results in a subset of patients with neuroendocrine tumors. This subgroup had an 83% clinical benefit rate and enhanced PFS. Upon the finding that oncolytic viruses synergize with checkpoint inhibitors and the discovery that SVV utilized a tumor selective receptor called TEM8 in 2017, we resurrected the SVV effort. SVV is currently being investigated by us to ascertain whether it is a suitable candidate for future clinical trials in combination with a checkpoint inhibitor. Checkpoint inhibitors work well in solid cancers that already have key immune infiltrating cells, such as T cells that are thought of as hot tumors. Unfortunately, these tumors are only a minority of the total. The majority of tumors are thought of as cold tumors. The key mechanism widely reported for the synergy of oncolytic viruses with checkpoint inhibitors involves the oncolytic virus's ability to selectively replicate in solid cancers and turn the cold tumors hot. Interestingly, in the prior SVV clinical trials, we observed in some patients an upregulation of the kind of immune molecules known to turn cold tumors hot, such as CXCL10 after SVV was administered. One last piece of background involves the receptor for SVV. TEM8 is abundantly expressed on the surface of tumor cells, but its expression is further upregulated when tumors progress and are invasive and metastatic. The expression of TEM8 in tumors correlates with poorer patient prognosis and survival in all indications examined to date. Here we are just showing breast and gastric cancer patients. One of the keys for us to determine whether SVV made cold tumors hot was to develop a cold tumor syngeneic murine model that expresses TEM8. A B16F10 derived syngeneic melanoma model called B78 was chosen as it is known to be immunologically cold. B78 cells did not express TEM8 and were resistant to SVV infection. Thus, a B78 cell line was engineered to express TEM8 and now became sensitive to SVV infection at low multiplicity infection. We also chose another cold syngeneic murine neuroblastoma model called N1E-115. This is already known to express TEM8 and is sensitive to SVV. We tested cohorts in each model, comparing no treatment to SVV, anti-PD-1, and SVV in combination with anti-PD-1. We further collected tumors at early and late time points in each group for RNA-seq analysis to determine whether SVV upregulated molecules known to be involved in the innate and adaptive immune response. In the B78 model, we saw a substantial slowing of the tumors, especially when SVV was combined with anti-PD-1. In fact, as shown in figure 4, the response rate of tumors treated with both SVV and anti-PD-1 was approximately 6-fold greater than either SVV or anti-PD-1 monotherapies. We also observed enhanced anti-tumor effects in the neuroblastoma model, which showed a 3-fold increase in response rate over the anti-PD-1 group when combined with SVV. These effects were corroborated by the RNA-seq analysis shown in figure 5, where we can see an antiviral response and activation of the innate immune response early after SVV administration. In the combination group, we see increases in gamma interferon signaling consistent with the induction of hot tumors. In summary, I'd just like to reiterate the key points from this poster. Thank you for your time.